Hi, my name is Christine Charvet. I'm an assistant professor at Delaware State University. I would like to thank the organizers for giving me the opportunity to present this talk entitled Structural and Transcriptional Variation linked to protracted human frontal cortex development. This work is in press and cerebral cortex, and Jasmine Hendy in my lab also will be presenting a poster on the subject. So we're interested in the study of human evolution and comparing brains, our brains, to those of other species, such as non-human primates and rodents. And we're really interested in the evolution of circuitry. Studying evolution of circuitry in humans is as much a study of how to study circuits as it is a study of identifying what about our brains deviates from that of other species. To study connections and axons, we have available tools called track tracers, which really showcase fine detailed structure as shown here in a mouse brain. But these methods are highly invasive and inaccessible to the study of humans. So in humans, we use diffusion MR tractography, which is a non-invasive, high-throughput method that allows us to visualize pathways throughout the entire brain. It's often used to better understand the neurobiological basis of a number of psychological disorders, but the tractography that we see here has not been without its uncertainties, so that moving forward, we're interested in integrating diffusion MR tractography with microscales of study to understand the evolution of development of frontal cortex circuitry in humans. We're particularly interested in integrating diffusion MR tractography with single-cell sequencing methods, which identify cell types as shown here in a t-SNE plot. And different cell types have stereotypical patterns of projections so that we could quantify cell populations across species, combined with comparative analysis of diffusion MR tractography to identify what is it about our brains that's different from that of other species. So I'll talk about our efforts integrating diffusion MR imaging and transcription to study the evolution of circuitry, and I'll also then talk about our investigations of the developmental mechanisms giving rise to change in circuitry. To that, we find corresponding ages across brain development because we need to find a solid foundation with which to compare brains. And then I'll talk about deviations in developmental programs using neuroimaging and transcription. So here's a human brain, diffusion MR scan that we see. So it's through the human brain. You can see that it shows these fibers coursing through the white matter of the brain. And we're going to integrate that with single-cell methylome sequencing studies. So as a first pass, we decided to compare humans to mice, and we chose mice because mice are a commonly studied model organism, and we know a lot about it so that we could get a lot of samples at different scales of study to understand what is it about the circuitry of a human that's different compared with a mouse. And so we noticed that humans had a whole lot of these pathways connecting the cortex that mice didn't. So we quantified these differences as shown here in a human and a mouse, and you can see that humans possess significantly more cortical pathways than mice in blue. And when we looked at the single-cell methylome sequencing studies by a study of Louis et al., we found that humans possess many more layer 3 neurons, or excitatory layer 3 neurons, in humans compared with mice. And we know that these are neurons that form cortical connections. That is, those are neurons that form connections with other cortical neurons. So that the findings from diffusion MR tractography, single-cell methylome sequencing, and other approaches that we use that I'm not mentioning here agree that humans possess more of these pathways connecting the cortex than mice do. So we were really interested in understanding the developmental mechanisms of change, and to that we needed to find corresponding time points across species to compare brain development between a human and mouse. So what we did is we extracted a RNA sequencing data set extracted from the frontal cortex of humans and mice at successive stages of development. We extracted when peaks in gene expression occur, and we collected peaks in gene expression, and then we collected other sort of transformations that occur during development. So here you can see the timing of these peaks in gene expression in humans and in mice plotted against days in conception, and those are shown in red. whereas these other transformations are in gray. And you can see that the inclusion of peaks in gene expression coupled with the timing of these other transformations does a good job at basically finding corresponding time points across species and finds corresponding time points up to 30 years of age, so that by capturing temporal variation in transcription, we can say how old is a human in mouse days throughout fetal stages of development and until adulthood. So we were really interested in understanding how development of pathway maturation might deviate in humans compared with mice. So we were really interested in looking at the temporal trajectories of 19 genes that are used as long-range markers. Here are some of these genes, 3 genes, NEFH, SITE2, VAMP1, and the normalized expression in humans in beige and mice in blue is plotted against a common developmental timeline shown here for mouse age expressed in days after conception. And it seems like the temporal trajectories of these genes in humans appears protracted compared with mice. So to capture this variation, we considered the coefficient of variation across all tested age ranges or only at late age ranges, with the assumption being that if the expression of these genes varies more so at late stages of development in humans compared with mice, this would indicate that the development of pathway maturation is protracted in humans compared with mice. So here's the coefficient of variation in humans in beige and mice in blue at all age ranges or only at late age ranges, and you can see that the coefficient of variation is significantly different in humans, or higher in humans than in mice, but only at late stages of development, suggesting that the expression of genes linked to these these projections that are connecting different cortical areas are protracted in humans compared with mice, even after controlling for variation in developmental schedules. So we sought to confirm this with other methods, and we know that the white matter of the cortex houses these axons that are connecting different cortical regions. So we measured the growth. We measured the growth of the frontal cortex in humans here on the top panel compared with mice on the bottom panel. Age in human is mapped onto mouse age, and what we see is that the frontal cortex continues to grow for longer in humans than in mice, even after controlling for variation in developmental schedules. So what we did is we captured epochs from these growth trajectories, asking when does the frontal cortex reach 100% of its adult stature, or 90%, or 80, or 70%, and we compared that to the timing in peaks in gene expression and anatomical and behavioral transformations. And you can see that these time points capturing frontal cortex growth cessation fall outside the 95% prediction intervals generated from the peaks in gene expression and anatomical and behavioral transformations. So the data from neuroimaging and transcription both show that pathway maturation in the cortex is protracted compared with mice, even after controlling for variation in developmental schedules. So these data show that we've been able to successfully integrate from macro to micro scales of organization to understand the evolution and development of frontal cortex circuitry. We've also been able to find corresponding time points across species. And so with that, I'd like to thank my lab members and let you know that we actually have a resource to find corresponding ages across species. It's called translatingtime.org. And I'd like to thank my funding sources.